Bismillahirrahmanirrahim. Assalamu alaikum. Dear fellows, today we are with the topic of gene regulation in bacteria. As you know that some structural genes, they encode messenger RNA which coded for proteins ultimately lead to a final product, functional proteins. This is a multi-step process that proceeds from transcription to translation to the post-translational mechanism. You are familiar with this mechanism, and this mechanism is termed as gene expression. Sometimes gene expression is regulated and sometimes gene expression is unregulated. What is meant by unregulated genes? The genes constant level of expression in all conditions. The genes which express themselves continuously, they are needed for the survival of bacteria and they are also called constitutive genes. But some genes are not unregulated, they are regulated. They are not continuously expressing themselves. They express only when they are needed. For example, in some metabolism, some proteins function in the metabolism of small molecules. They only produced when, for example, the enzymes needed for the metabolism of particular sugar, and the enzyme required only when bacterium exposed to such sugar in the environment. If that sugar is present in the environment, they produce an enzyme for the metabolism of that sugar. And if that sugar is not present in their environment, they will not produce their enzyme for the metabolism of that sugar. Second condition, response to environment. Some proteins help in response to certain environmental stress, and these proteins required only in the stress condition and not required in rest condition. Third, cell division. Some proteins needed only during the cell division and are not needed in rest phase. So when they are needed, then they are produced by this process. So this is called gene regulation. It is the phenomena in which the level of gene expression can vary under different conditions. So this is the gene regulation, that some genes only express themselves when that protein is required, and cell do not waste energy for the making of protein which are not required. This is the advantage of gene regulation, that cell saves its energy. So we will explain the basic mechanism of gene regulation in bacteria. So I am going to explain the basic mechanism of gene regulation in bacteria. The most common way of regulating the expression of gene is influenced by the rate of transcription. We can say that expression of gene can be increased or can be decreased. We can also say that rate of transcription can be increased or it can be decreased. In most cases, transcriptional regulation is involved by the effect of regulatory proteins. The regulatory proteins binds to the DNA and affect the transcriptional process. There are two types of regulatory proteins, repressor and activator. Repressor are those regulatory proteins that bind to DNA and can inhibit transcription. Means the gene is transcribing messenger RNA, but when the repressor protein is bind to the promoter region of gene, it will stop the transcription and messenger RNA synthesis stop. This control is called negative control, mean a repressor protein that binds to DNA and inhibit the transcription. Second regulatory protein is activator protein. This is a regulatory protein that increases the rate of transcription. When this activator protein binds to the gene, to the promoter region of gene, after binding the protein, the gene starts transcription and the gene starts to synthesize messenger RNA. This control is called positive control. Apart from regulatory proteins, there are small effector molecules which play critical role in expression of gene, in regulation of gene. These small effector molecules do not bind directly with DNA. They bind to the regulatory proteins, either to repressor or to an activator, and causes conformational changes in regulatory proteins, influencing whether or not the proteins can bind to DNA. There are three types of small effector molecules: inducer, co-repressor and inhibitor. Inducer, we will see them one by one. Inducer is a small effector molecule that causes transcription to increase. Inducer is a molecule that increases the transcription. It increases the transcription by two ways. First, in the absence of inducer, repressor protein binds to DNA and stop transcription. In the presence of inducer, repressor proteins do not bind to the gene and genes start to transcribe messenger RNA. Attachment of inducer inhibit the repressor protein to attach to DNA. Similarly, in the absence of inducer molecule, activator protein do not bind to the DNA and DNA do not synthesize messenger RNA. But in the presence of inducer, activator protein attach to the DNA and DNA starts to synthesize messenger RNA. In these two procedures are involved in increasing the transcription. The second small effector molecule is co-repressor. Co-repressor is a small molecule that binds to repressor protein, causing the protein bind to DNA. In the absence of co-repressor, repressor protein will not bind to the gene and gene synthesize messenger RNA. But in the presence of co-repressor, the repressor protein will bind to gene and stop the transcription process. Messenger RNA synthesis stops. This is a co-repressor molecule that binds to repressor protein and which activate the repressor protein, and repressor protein bind to the gene which stop the transcription. The third small effector molecule is inhibitor. Inhibitor is a molecule that binds to activator protein and prevents it from binding to DNA. It means this inhibitor molecule binds to activator protein and it releases the activator protein from the DNA. The activator protein which increase the rate of transcription. In the absence of inhibitor, activator will attach to DNA and the gene or DNA transcribe messenger RNA. But when this inhibitor molecule is attached to activator protein, it causes conformational change in the activator protein and activator protein releases from the DNA and transcription process will stop. So in this way, this inhibitor molecule reduces the rate of transcription. These are three small effector molecules: inducer, co-repressor and inhibitor. Inducer increase the rate of transcription and co-repressor or inhibitor decrease the rate of transcription. This was the overview of gene expression in bacteria. There are two regulatory proteins, activator and repressor, and there are three small regulator molecules that affect those proteins which regulate the rate of transcription. In next video, I will come with a suitable example of gene regulation in bacteria.